The overall goal of this procedure is to evaluate the anti-tumor immunity of a cancer stem cell or CSC-based dendritic cell or DC vaccine in an immunocompetent host. This is accomplished by first isolating aldofluor-positive aldehyde dehydrogenase or ALDH-high CSCs from heterogeneous tumor cells. In the second step, CSC DCs are isolated and then pulsed with lysate for the vaccine. Next, the CSC lysate pulsed DC or TPDC vaccine is administered to normal syngeneic immunocompetent hosts. Then, in the final step, the protective antitumor efficacy of the CSC TPDC vaccine is assessed. Ultimately, ELISAs can be performed to evaluate the CSC TPDC vaccine-induced anti-CSC immunity by host T cells and antibodies.
This project brings together two of the most exciting areas of cancer research, the area of cancer stem cells and the area of tumor immunotherapy. Our lab was among the first to discover cancer stem cells in solid tumors. These are the seeds of the cancer that drive its growth as well as metastasis. What you'll see today is a new approach using immunotherapy to specifically target these cancer stem cells. We utilize a particular marker called aldehyde dehydrogenase, which can be detected by the aldofluor assay. You will see Drs. Lin Lu and Huimin Tao working along with Dr. Qiao Li. We hope that by targeting these cancer stem